braving the elements that are coming in. I hope to make the information and the learning worth it. So first announcement, there's a biology colloquium this week, Thursday at noon in CST 300 this week, upstairs in the big room. Emmy Smith is going to be talking about the recent event in human genome engineering. A Chinese scientist used the CRISPR-Cas technology to edit viable human embryos, and those children have been born. So, discussion of the ethics, not just the biology, so she's inviting people from all over campus to come and participate in this discussion of the ethics of genome editing. Very timely topic. That's Thursday at noon. Let's see. Let's look at the Moodle and just see what you can see here on the Moodle. So, you can see I've loaded PowerPoints. The problem set is up there. My answers to the problem set are now posted. I haven't graded yours yet, but my answers are there, and you can look at this, compare mine to yours. The lab worksheet for this week, so we're in week 3 now, so the Tuesday group is going to do this lab today. The Thursday lab did this last week. And then, so that the worksheet is available now, but the Tuesday lab will work with it today in lab. And my answers will be posted as soon as I unhide it from you. Today's PowerPoint will be up there as soon as I reveal it. Not yet. And next week's worksheet is also posted, but not the answers yet. Okay, I have to make an announcement about Quiz 1, because when I worked up the schedule, I wasn't thinking about the size of the class and the 3 lab phenomenon. And so I realized that in the schedule, the Quiz 1 is the deadline is Sunday in the schedule. But I realized, kind of late in the game, that means that the Thursday lab will have had 2 times with the cadaver, whereas the Tuesday labs would only have had 1. And that just didn't seem fair. So I'm postponing Quiz 1. So Quiz 1 is not due on Sunday anymore. It is now due on Wednesday, after the Tuesday lab has had a chance to do the worksheets with each other, with me in the room, etc. So it will be available on Friday at the desk, and you turn it in by Wednesday, not by Sunday. Aiden?
So, are there things on the quiz that are not in the cadaver of the lab, but just on the...
Yeah, there are things on the quiz that are not in the lab.
That were just from the
But look on the key concepts list, okay, so the list that I gave you today, the key concepts list for the quiz is a subset, mostly, of the key concepts for the whole section. This is your study guide for the quiz.
Okay.
So, for example, we're not looking at nerve cell biology on the cadaver, but that could be on the quiz. Anatomical terms, muscles. We're probably not going to see the muscles of the lower arm on the cadaver yet, but that will be on the quiz. Bones, we're not even close to seeing bones, very many of them, but that will be on the quiz. So the cadaver is helpful, but the quiz is not limited to things we see on the cadaver.
Is that changing quiz 2?
Pardon?
Is that changing quiz 2?
No. That will change when quiz 2 will be due as well. Quiz 2 will be due on the Wednesday following the Sunday that is listed in the schedule. I think it's the Wednesday before mini break. Mini break is so early. So here's your key concepts. This is your study guide for the quiz, and I'll post another one for quiz 2. The craniotomy and brain removal will be done on Sunday. That's not being postponed to Wednesday. That will be on Sunday afternoon, 2:30 to 5 o'clock. optional. It's really interesting, it's amazing, but it's optional. And you can come for some or all of the time, and you can participate or just watch. It's really just sort of an open house. To students, not to anybody. Questions about any of these announcements? Okay, well then let's get to work and talk about synaptic and action potentials. So let's start with synaptic potentials. We'll come back and talk about the mechanisms of synaptic release of neurotransmitter later. But right now we're focusing on a synapse. We've got a synaptic terminal, this little dot here, a presynaptic terminal. Some books and people call it presynaptic bouton in French. Presynaptic button, presynaptic terminal. This is where neurotransmitters are going to be packaged and released by exocytosis. The receiving cell has a postsynaptic specialization full of receptors to the neurotransmitter that is released by the presynaptic terminal. So this is a axosomatic synapse. Axosomatic synapse. A typical synapse would be an axodendritic synapse. There can also be axonic synapses. And frankly, there are a wide variety of other ones too that we don't need to focus on. We're focusing now on the electrophysiological response of the postsynaptic cell. We'll come back to the release of neurotransmitter later, but now just remember, neurotransmitter is released and it causes a postsynaptic potential, a change in the membrane potential of the postsynaptic neuron. So here's the words that describe it. Synaptic potentials are small, local changes in the membrane potential that spread locally, limited locally, and passively, not by a simple diffusion of electric charge. That's passive spread. They summate. So if all four of these terminals release neurotransmitter, you would get a larger postsynaptic response than if only one of them did. That would be spatial summation. If this synaptic terminal is triggered 10 times in a row, it'll release more neurotransmitter than if it only is triggered once, and that's temporal summation. And the more neurotransmitter from the 10 triggers would cause temporal summation of the postsynaptic potential. So we're focusing on the potential of the postsynaptic cell. Then later today, we will talk about action potentials that are large changes in membrane potential, regenerative, I'll define that, propagating, we'll talk more about that on Thursday, changes in the membrane potential. So we're not talking about this just yet, we're talking about this. These postsynaptic potentials can either be depolarizations or hyperpolarizations, excitatory or inhibitory postsynaptic potentials. So at an excitatory synapse, so any given cell, so we're just looking at the release of information from this cell to this cell, one cell. And each of these synapses of this cell on this cell would either be excitatory or excitatory or inhibitory. this cell is going to release some type of neurotransmitter, and that was determined during the development of this cell. The 1,000 of other cells that also make synapses on this cell will have other neurotransmitters that were determined by the development of those other cells. And those other cells, those other neurotransmitters, might be inhibitory or excitatory, depending on the neurotransmitter. So, one more point I want to make from the previous slide. This neurotransmitter, let's just say it's excitatory. All of these from the same neuron are excitatory. And they create a specialization in the postsynaptic cell during development that recruits excitatory neurotransmitter receptors to the location of the synapse, to the postsynaptic specialization. Inhibit a different neuron, one of the thousands of other different neurons that might be releasing inhibitory neurotransmitters, there would be inhibitory neurotransmitter receptors at those synapses. So the cells are specialized by what neurotransmitter they release, and there are specializations during development that localize the correct types of receptors at the correct types of synapses. So we're not going to talk too much about developmental neurobiology. That's a whole course by itself. Leah?
So neurons usually are excitatory or inhibitory, usually.
Yes. Neurotransmitters are either excitatory or inhibitory, usually.
So neurotransmitters.
Yeah.
Neurons are both. Neurons can release either.
No, one neuron. So neurons can release either, but one neuron either releases one or the other. So here's a cartoon of an excitatory synapse with an excitatory neurotransmitter receptor. At rest, the cell is negative on the inside. You've now explored quantitatively through the problem set the features, the factors that lead to how negative this cell is at rest. That was in your problem set. If after you've looked at my answers and compared them to your answers to the problem set, you have questions about the quantification of it, I'm happy to meet with you anytime and clarify. At rest, negative on the inside. When this receptor is activated by its neurotransmitter and opened by the conformational changes triggered by the binding of the neurotransmitter to the receptor, an excitatory receptor allows sodium to flow in. The sodium flowing in reduces the negativity on the inside because we're bringing more cations in, and that's a depolarization excitatory response. On the problem set, the example that I used for this was the nicotinic acetylcholine receptors. They are acetylcholine activated ion channels that are not selective for sodium over potassium, but because the inside is negative, we get more sodium influx than potassium efflux. More sodium influx creates that depolarization that you should have seen in that answer to that problem set, answer to that question. So with their acetylcholine is usually an excitatory neurotransmitter. Glutamate is another excitatory neurotransmitter that opens ion channels that are sodium permeable. Some of them are calcium permeable. We'll talk specifically about receptors later. That's two examples of excitatory neurotransmitters, acetylcholine and glutamate, and ionotropic, that is to say ion channel-based receptors to those excitatory neurotransmitters that cause a depolarization. At an inhibitory synapse, a different neurotransmitter would activate a different receptor. These two receptors look close to the same. This one's got a little jagged edge, this one doesn't. This is a different receptor because it's a receptor to a different neurotransmitter, an inhibitory neurotransmitter. At rest we're negative, just like we were before. Activation of this inhibitory neurotransmitter by activation of this inhibitory receptor by its neurotransmitter activates a channel that is chloride, not sodium, permeable. Chloride flows in down its electrochemical gradient. Even though the negative is inside, there is still, because the chloride concentration is higher on the outside than on the inside, chloride still flows in, makes it more negative on the inside, and that tends to either maintain the polarization or even hyperpolarize the neuron. And you saw this with the GABA receptor question on the problem set. Now, if the chloride concentration is not big enough to allow chloride to flow in when these channels are open, if, say, the intracellular chloride was not so small, then opening this receptor would allow chloride to flow out down its electrochemical gradient, and that would have been a depolarizing response. And we saw this on the problem set too, in the developing interneuron question on the problem set. But usually, opening of ligand-gated chloride channels allows chloride to flow in and polarizes or maintains polarity or hyperpolarizes the neuron, making it an inhibitory neurotransmitter. GABA, glycine are typical inhibitory neurotransmitters in the nervous system. Shall I?
So if we are asked, like in a quiz or something without numbers about the concentration gradient, do we focus on the chemical, not the electrochemical gradient of chloride?
I will not ask you to do calculations on a quiz.
But it's just like to understand where the chloride flows, or would you ask the question similar to the problem set, where you say it would do like the opposite of what it usually
Yeah,
does?
I think, so what I would say is typically, in a typical situation, which way is chloride flowing?
Okay.
If you know that it's a inhibitory neurotransmitter, and you know it's causing an inhibitory response, that is to say making the inside more negative, you would know that chloride, and you know it's a chloride channel, you would know that chloride has to be flowing in. So I might ask you to put that all together, either, knowing the typical situation, or even challenge you to say, okay, I've just screwed up this cell and I've collapsed the chloride gradient completely. The chloride concentration on the inside is the same as the outside. Now what happens when GABA is added? I would ask you to solve that. I could ask you to solve that problem. Questions? Are all inhibitory receptors chloride channels? No. If a receptor were coupled to a potassium channel, that could also be inhibitory, right? Because remember, potassium flows out down its concentration gradient. Increasing the potassium permeability could also be an inhibitory response. But this is illustrating the chloride side of that story. All of the excitatory responses are influx of either sodium or calcium. All of the inhibitory responses are either influx of chloride or efflux of potassium. And I just said all, and that makes me very nervous, but I'll stand by it. So, if we look at, here now, we're looking at the same information, but not in a cartooned form of plotting the membrane potential as a function of time. Here's the membrane potential at rest in this cartoon, minus 70 millimoles. We stimulate the presynaptic neuron. So it releases neurotransmitter, and in this case, the presynaptic neuron caused an excitatory postsynaptic potential, EPSP, a small, local depolarization due to sodium influx. Here, a different neuron was stimulated. Resting membrane potential stimulated the neuron, caused a small hyperpolarization and inhibitory postsynaptic potential. A small local inhibitory response. A small local hyperpolarization due to chloride influx. These responses are small and local. They spread and they summate. That is to say, they are graded. So there's two types of potentials that we'll talk about in this class. Synaptic potentials, which are graded, and action potentials, which are, quote, all or none. Both of these, so what does graded mean? Graded just means it is variable. There are big ones and there are little ones. You get 100 or you get a 90. That's a graded response. You don't, this course is not graded, pass, fail. That would be a all or none response. So, synaptic potentials are graded in the sense that a stronger stimulus causing either temporal or spatial summation gives a larger response. That's what it means for them to be graded. They are small, they are graded, and they spread locally. So if we measure it at the stimulus site, we get a certain size response. And if we measure it a millimeter, that's pretty far away on a synaptic scale. If we measure it away from the release site, the response is smaller because the response has diffused away from it, right? So this is passive local spread of a excitatory response. Right near the receptor, the depolarization is great. Further away from the receptor, the depolarization is smaller because of the spread. If I walk out into the hall and start talking at the same volume, the volume in this room will go down because I'm further away from it. Sound is a graded phenomenon. There are equations, so physiologists, biophysicists love to make equations whenever possible. So if you want to be biophysical, I'll be happy to discuss this spread with you in biophysical terms, but you don't need to know it. So, synaptic potentials are small, local, graded, and they summate. The 10,000 of them add up or subtract off from each other, depending on that. So here is a soma. You can see 1, 2, 3, 4 truncated dendrites, but they would be going out here. Here is the axon. Remember, the segment of the axon that is closest to the cell body is called the axon hillock, or the initial segment, or the initiation zone of the axon. It's got 3 synonyms. Now, so the question is, do all synapses influence the creation, the initiation of action potentials equally? That's a question. Anybody want to answer my challenge question? Kendra?
I'm going to say that because it depends on how much of the ion flows into that cell.
Okay.
That dictates how much summation the cell happens.
Okay. So Kendra's saying no. Anybody want to amplify on what Kendra is saying? That's the correct answer. So let me just help you be confident and to say it either in another way or say something else that builds on that.
Possibly because they spread, if they're closer to the axon, I feel like they could have more of a influence.
Sure. This synapse, this one synapse, is going to have much less influence on the axon hillock than this synapse. So no, they're not all the same. Even if they were all excited, obviously if the inhibitories have a different influence than the excitatories, that's one answer. And the other is that the distal synapses have a smaller impact on the hillock, on the initiation zone, than the proximal synapses. So placement of the synapse matters because of the spread phenomenon. So it might take 10 synaptic potentials here, 10 EPSPs, to create the same depolarization here that one EPSP here causes. Just making up some numbers. So we've talked about summation, and so here's an easy cartoon version to see summation. So we have an excitatory, so the green neuron, color-coded, the green neuron releases an excitatory neurotransmitter. The red neuron releases an inhibitory neurotransmitter. Let's just say glutamate and GABA. We are recording the membrane potential of the postsynaptic neuron. Recording the membrane potential as a function of time. If we stimulate the presynaptic neuron, we get a response, which decays because the neurotransmitter is cleared from the synapse. So neurotransmitter goes up, EPSP, neurotransmitter is cleared, EPSP goes away. Do it again, same thing. Do them close together, the second response is bigger than the first response due to temporal summation. Stimulate both A neuron and B neuron, and the excitatory response is bigger still. Or bigger than just A alone because of spatial summation. Neither A nor B alone was big enough to cause the membrane potential to get to threshold, the voltage at which the neuron, the voltage at which the initiation zone triggers an action potential. So these are sub-threshold EPSPs. All of them are sub-threshold EPSPs. But if we do both temporal and spatial summation, that's what's going on here, we can get this neuron to threshold at which we can get the spike initiation zone above the threshold, the voltage necessary, the depolarization necessary to trigger the action potential that we're going to be talking about in a few minutes. So one synapse doesn't do it by itself, but two synapses doing spatial and temporal summation might. It's probably an exaggeration. You probably need more than two, but you can see the point here is what the point here is. And summation can be negative. So stimulating A and C at the same time creates no response. Negative summation, subtraction. We don't call it subtraction, we call it negative summation. So now let's talk about action potential. Question?
Sorry, yeah,
Yeah.
on the previous slide, so, where the peak is.
This here?
Yeah.
That's the action potential.
Yeah. Does that mean that A and B are no longer being fired, or does that mean that C is being fired?
Here?
Yeah.
At here, let's see. So A and B have both been stimulated, but so A was stimulated here and here. B was stimulated here and here. So here, I guess you'd say A and B are still active, but not C.
Yeah.
So now we're going to talk about this phenomenon, the action potential. Action potential. So action potentials are called all or none. They are you either have one or you don't. Now, that's somewhat of a, I just want to clarify that it's not completely correct to call them all or none, because here's an action potential, and here's an action potential, and they are not exactly the same size. But we still simplify it by calling action potentials all or none. They are not considered graded potentials. And we'll talk about why they are not graded in a minute. So here's a whole string of action potentials that are all pretty much the same size.
Isn't the reaching to the action potential is the all or none? The reaching to action potential is
Yeah,
the all or
the
none?
amplitude, that's right, the amplitude of action potentials is all or none. The frequency is modulated. So that's another important point. It's not the amplitude of action potentials that carries information, because the amplitudes are all pretty much the same. It's the frequency, low, medium, high, it's the frequency of action potentials that carries information in the nervous system. This is the response to a small depolarization. Here's a different response to a small depolarization showing the, showing this sort of double stutter here. And here's a response to here's a different response to a very slightly different, oh no, this one, this is a bigger stimulus. Bigger stimulus, higher frequency response. Action potentials, so the information is carried by action potentials not in their amplitude, but in their frequency. That's not true of synaptic potentials. Synaptic potentials being graded, information is carried in their amplitude as well as their frequency. So this is information in our brain. When you are seeing me, the light that is reflecting off of me and into your eyeballs is transformed by your retina into electrical information. Light doesn't go into your brain to say, oh, there's Bob. Electrical information goes into your brain, and your brain then turns that electrical information into, oh, there's Bob. Same thing with sound. The sound isn't going into your brain. Well, maybe it is, but that doesn't matter because your brain doesn't detect sound. Your inner ears detect sound, and then electrical information is carried to your brain, and that says, oh, Bob is talking. I better listen. So the point is, we'll talk about how your retina converts light into electrical signals. We'll talk about how your cochlea turns sound into electrical signals. That's a big part of this course. But for now, we're just talking about the action potentials. So let's talk about the jargon, the language of action potentials. So we've got a resting membrane potential, minus 70 or something. Depends on how many potassium channels are open or not, right? We did that on the problem set. If we opened more potassium channels, we could make the resting membrane potential more negative. Here's the equilibrium potential for potassium. So in this case, it was minus 90. In your problem set, I think the correct answer was minus 97.7 or something. Here's the equilibrium potential for sodium. In this case, it's plus 60. In your problem set, it was plus 66.6. Here's the stimulus, the EPSP, probably a summated EPSP, that triggered this neuron to fire its action potential. So this EPSP got to threshold and triggered an action potential. An action potential is a large, transient depolarization, very short-lived, only about 1 millisecond, maybe 2 in duration. Many tens of millivolts, large, much larger than an EPSP. So this is a summated EPSP. A single EPSP would probably be this size on the scale. A large, short-lived depolarization that can propagate. We'll talk about propagation on Thursday. Large depolarization that then repolarizes and sometimes, often, has an after hyperpolarization. So some of the jargon, threshold, depolarizing phase, repolarizing phase, after hyperpolarization. The overshoot is the voltage above 0. in your problem set, the overshoot of that, when I asked you to solve the problem related to the action potential, was 45 millivolts. The amplitude is the overshoot plus the voltage below zero that it started. So if this is minus 70, and this is plus 40, that would be an amplitude of 110 millivolts. There are two types of channels that are most important for understanding the action potential. The voltage-gated sodium channels and the voltage-gated potassium channels. The voltage-gated sodium channels are responsible for the depolarization, and the voltage-gated potassium channels are responsible for the repolarization and the after hyperpolarization. Voltage-gated sodium channels are fast. Voltage-gated potassium channels are slower. It's just part of their biophysical characteristics that voltage-gated sodium channels respond to the voltage at reaching threshold more quickly, faster than the voltage-gated potassium channels do. So that's why the voltage-gated sodium channels open first, and then the voltage-gated potassium channels open. Which way is sodium flowing through voltage-gated sodium channels? Inward. Which way is potassium flowing later during the red phase? Outward. So despite the fact that this is going up, this is influx, and this is efflux. Mike? They do have approximately the same voltage sensitivity, but one is faster and one is slower. Their voltage dependence are very similar.
Is the sodium potassium pump still going?
The sodium potassium pump is still going. It's maintained. So despite the fact that sodium has flowed in and potassium has flowed out, the concentration gradients are not much changed. Negligible change, because the amount of sodium flowing in and the amount of potassium flowing out necessary to make this signal is small, and the pump can keep operating. So let's talk about voltage-gated sodium channels. This is getting to that word regenerative that I mentioned in the description of action potentials. So here's the starting point. A small local depolarization. A small local depolarization. The stimulus, the thing that gets the ball rolling. A small local depolarization opens voltage-gated sodium channels. That's what they respond to. They have a voltage sensor. They sense the voltage, and they undergo a conformational change in response to a depolarization. So a small local depolarization opens some of the voltage-gated sodium channels. Conformational change is embedded in this arrow, opens the channel, sodium flows in. The influx of sodium causes more depolarization. More depolarization opens more voltage-gated sodium channels, causes more of them to undergo opening conformational changes, more influx of sodium, more depolarization. We go around this cycle in 100 microseconds. We open all of the available voltage-gated sodium channels. And that's why it's all or none. A small local depolarization triggers a cycle of activity that in 100 microseconds or so opens all of the channels, all of them. And so it's all. We either, if we didn't, so that's the all part of the all or none. If this depolarization was too small and didn't get to threshold, it wouldn't cause an action potential at all. And that's the none part of the all or none. So if we get to threshold, we open them all, and we get a full-blown action potential because of this positive feedback loop that is very fast. And it's fast because the voltage-gated sodium channels have fast conformational changes. 100 microseconds. So we have just gone from here to here. We have opened all of the voltage-gated sodium channels, and the membrane potential has gone way positive, moving towards the equilibrium potential for sodium. Moving towards positive 66.6, but not getting all the way there. It's not getting all the way there because immediately after voltage-gated sodium channels open, they inactivate. So what was the slide that was? Okay, I've said these words. After voltage-gated sodium channels open and do their positive feedback loop, about a millisecond into the afterwards, they undergo a different conformational change that causes them to inactivate. And so, even as they are shooting the membrane potential up, some of them are starting to inactivate, reducing the sodium permeability. So the membrane potential doesn't get all the way to E and A, to plus 66. It gets to plus 20, plus 30, plus 40, plus 50, plus 45, depending on the node. But whatever it is, it's all of the available sodium channels. Different cells can have different amplitudes, but one cell has a typical all situation. Okay? So after they all open, they all, with some kinetics, random kinetics, inactivate. So the inactivation of the voltage-gated sodium channels terminates that the factor that is causing this large depolarization. And so now the membrane potential can and does repolarize, because remember, we have all those resting potassium channels. So, sorry, I keep going back and forth. When the sodium channels inactivate, the resting potassium channels and now the increasing voltage-gated potassium channels bring the membrane potential back down towards the equilibrium potential for potassium. So inactivation, inactivated channels are not permeable to sodium. So let's talk about the states. This is now a state diagram of sodium channels. At the resting membrane potential, so if you look at this equilibrium here, most of the channels are closed. The inactivated ones have been able to recover to the closed state, and there are very few open channels. So at resting membrane potential, channels, voltage-gated sodium channels are closed. What's the difference between a closed voltage-gated sodium channel and an open voltage-gated sodium channel? Let's do an easy one first. Yeah, conformational change, closed to open. What's the difference between a closed voltage-gated sodium channel and an inactivated voltage-gated sodium channel?
A closed one can possibly open from the activated.
Exactly. A closed voltage-gated sodium channel can transition to the open state, where a voltage-gated inactivated, where an inactivated voltage-gated sodium channel has no connection to, there's no arrow going back this way, and there's no arrow going to the open state without the first closing. So an inactivated voltage-gated sodium channel is not permeable, but it is not closed, because I try to use the word closed to reserve for a closed channel that can open. An inactivated sodium channel is not permeable, and it can't open. It's like it's a closed state from which you can't open, whereas you have a closed state from which you can open. So, Aiden.
I think I've seen like pictures of something like a plug or something that comes in and like
There is something very much like a plug that causes the inactivated state.
Okay.
It's part of the protein. Transitions into the pore and plugs it. When the membrane is depolarized, closed channels can open and then they inactivate. This is a fast kinetics and this is slower kinetics. Only during repolarization, so during depolarization, channels go from closed to open, and then they inactivate. And if we stayed at a depolarizing voltage, they would stay inactivated. Only the transition from inactivated to closed requires repolarization. And it's just a phenomenon of the channel that this transition requires a negative membrane potential. So all of these kinetic steps, and you've seen some kinetics, we did some kinetics in self-phys, you've probably done some kinetics in chemistry. All of these kinetic steps are voltage dependent. This one is accelerated by depolarization. This one only happens when the channel is open. This one is accelerated by repolarization. Voltage dependent kinetics, voltage dependent gating of the channel. So the channels open and then inactivate. While the voltage-gated sodium channels are opening and depolarizing, the depolarization also opens voltage-gated potassium channels, but with slower kinetics, even slower than the inactivation kinetics of voltage-gated sodium channels. Now more potassium efflux, more repolarization. And so this accelerates or amplifies the repolarization phase. And this is why we get the after hyperpolarization. Voltage-gated sodium channels activating, voltage-gated sodium channels inactivating, and voltage-gated potassium channels opening, causing repolarization. At this point, voltage-gated sodium channels are still mostly inactivated because and voltage-gated potassium channels are mostly open, and we have a hyperpolarization. It takes a few milliseconds for the channels to undergo the conformational change for them to undergo the conformational change from inactivated to closed. So here, the sodium channels are still mostly inactivated, and the potassium channels are mostly activated, and we get a hyperpolarization. Then as the give it some time, the voltage-gated potassium channels close, and the channel, the voltage-gated sodium channels, recover from inactivation, and we get back to the resting state from which we could fire another action catcher. So let's take a break, and I've got some challenge questions and some clicker questions that will allow us to work with this. Let's take a short break, and then got a series of questions.